Hi everyone, I'm Pendy from Multimedia University, Malaysia. This work was in collaboration with Benjamin Vakif University, Turkey. Thank you for joining my section. My talk is about studying the protein sequence change dynamic of Primate erythroparvovirus 1. The virus is generally known as parvovirus B19 or B19V and is a human viral pathogen that commonly infects children, causing erythema infectiosum. Herein, we report the proteome sequence diversity dynamic of B19V. All reported protein sequences of the virus from human hosts were retrieved from the NCBI Entrust protein and virus databases. A total of 6,562 protein sequences were collected, deduplicated by use of CD HIT, processed into separate protein by use of BLAST-P, and aligned individually by use of cluster omega. Table 1 here lists the number of redundant and non-redundant sequences of the 6 proteins coded by the virus. One could surmise that the virus has been understudied given the small number of sequences for each protein. Thus, only 3 of the 6 proteins, VP1, VP2, and NS1, which have sufficient sample size of more than 30, were further analyzed. The analysis were carried out on the alignment of the proteins, a sample alignment shown in Figure 1. Channel entropy calculation and quantitative pattern analysis of sequence diversity motif were carried out for each of the overlapping non-normal positions of the proteins. The motif includes the index and its variants, which are subgrouped into major, minor, and unique, which I will explain further later. Coming back to the measuring entropy of the overlapping non-normal positions, Figure 2 here provides the overview of the entropy and the corresponding total variance. Entropy values are on the left y-axis, whereas total variance are on the right y-axis. Higher entropy values indicates higher diversity, and the lowest entropy value of 0 indicates complete conservation. Total variance is defined as simply 100% minus the incidence or frequency of the most predominant sequence, which we refer to as the index. In this plot, we have the entropy of NS1 shown, and here for BP1 and BP2. Do note that BP2 is a subset of BP1. The entropy pattern between the proteins are different, and the positions were generally of low entropy value, interspersed by several peak positions. The highest peak of 2.8 was observed in the protein NS1. For benchmark, this line indicates the highest non-normal entropy values that we observe from our various analyses of viruses. The peak absolute entropy of 9.2 and total variance of 98% was observed for HIV-1 clade B. In general, the B19V proteins have a low mean entropy of roughly 0.5, with only 9 positions exhibiting total variance close to 58%, indicating high conservation. Figure 3 provides a scatter plot to better understand the relationship between entropy and total variance. Each dot represents a non-normal position. A positive correlation was observed with a wider spread in total variance with increasing entropy or diversity, starting from entropy value of 0.5 onwards. Given this, we then went on to investigate the diversity motif that comprised the total variance, namely the major, minor, and unique variants. The distribution of index, which is the predominant sequence and its variance motif across the individual protein non-normal positions, illustrated a distinctive pattern of sequence change dynamic. This is shown in Figure 4 and 5. Referring to Figure 4, the incidence of the index range from 100%, which is completely conserved, to as low as 42%. The major variant, which is the second most predominant sequence at a given non-normal position exhibited a rough pyramidal pattern. Minor variant, the non-normal sequences that were observed more than once but of lesser frequency than the major variant, was strikingly absent in roughly 77% of the non-normal positions. Thus, most of the protein non-normal positions lack minor variant. Notably, unique variants, non-normal sequences which were observed only once in the alignment, were seen for nearly all the protein non-normal positions. Nonotype, defined as the distinct non-normal as a given position, were all below 25%. The violin plots here in Figure 4 show the distribution of the incidence value for each motif. It provides the overall patterns for the viral proteome. In other words, the proteins analyzed together. When analyzes the protein individually, the same trend was observed as shown in Figure 5. In Figure 6, we can see that all the non-normal positions of the protein study could be classified into several categories of conservation level, namely complete conserve with index incidence equal to 100%, highly conserved, defined as index incidence in between 100% to 90%, mixed variable, defined as index incidence in between 90% to 20%. B19 proteins analyzed were devoid of highly diverse and extremely diverse, index incidence lesser than 20. Thus, it could be said that primate erythroparvovirus 1 protein viral proteome is highly conserved, with roughly 73% of the proteome position comprising the protein study were complete or highly conserved. In summary, this study provides insight to the primate erythroparvovirus 1 protein sequence diversity dynamic. A limitation is that the study could only be done to the 3 proteins NS1, VP1, and VP2. We hope that this could be repeated in the future, then more sequences are available for the remaining 3 other proteins. Thank you.